did. They were among the most important groups in terms of working out how to get this to work in eukaryotic cells and to use it for gene editing. They weren't the ones who discovered CRISPR. CRISPR was discovered in bacteria, and we were just talking about this, that there's some discussion about who should get the credit for actually discovering CRISPR, but that's beyond what we need to do today. But these two got the Nobel Prize for working out how to make it happen in eukaryotic cells. So Jennifer Doudna and Emmanuel Charpentier. I always like the fact that Jennifer Doudna's last name ends D-N-A. All right, so CRISPR is a natural phenomenon in bacteria. It seems to be utilized by essentially all bacteria that have been looked at. Of course, not all bacteria have been looked at, so I can't say all bacteria do it, but certainly a lot of bacteria do it. So what I need to do first is to explain to you how bacteria use this. And once we've done that and we understand that, then I can explain how we can transplant that into eukaryotes and get eukaryotes to do it. So we're going to talk about it in bacteria first, which is the natural system. Now, CRISPR is not the first gene editing technology to come along. So I always like to say that gene editing started when the first humans started doing controlled breeding of animals and plants. So the idea of engineering organisms has been around a long time, right? So the difference with molecular approaches is that instead of modifying animals by breeding, you're doing it faster with molecular techniques. But both methods ultimately end up changing the DNA, right? So fundamentally, they're not all that different. It's just one is slow and one is fast, right? One is hard to control and one is easier to control. But breeding has been incredibly successful. All you have to do is look at the difference between Chihuahuas and Great Danes to see that breeding can be incredibly amazing at modifying organisms. Okay, so CRISPR genome engineering, a huge breakthrough because it made genome editing really efficient and simple and inexpensive as opposed to complicated, inefficient, and costly. So there were methods for doing this before, but they were not practical for most labs to do. It was just too complicated, too expensive. And so I could waste a lot of time telling you about the zinc finger technologies and Talon technologies, but none of those things are used anymore. So it doesn't make sense for me to talk about them, so I'm not going to waste our time doing that. We're going to go straight into CRISPR. Okay. So this is an artistic rendition of CRISPR happening. This would be your guide RNA. This would be the part of the genome that's being edited. So we'll come to that. Okay. So if we go back to this idea of breeding I mentioned yesterday, that one way to try to speed things up was to use various ways of introducing mutations into organisms quickly using radiation or chemicals. And so that was done for a while, and I think I said last time that red grapefruit and seedless watermelon were two products of radiation-induced mutations. Bryce pointed out that's not right. It's true for red grapefruit, but not for watermelon. But a lot of the important rice and wheat and barley varieties used today were developed many years ago by mutating with radiation to introduce changes. Now, a big drawback of that approach is that if you're using radiation or chemicals, you can't control where the mutations occur. And when you mutate an organism like that, you're typically going to introduce many mutations. You might get the one you want, but you're also going to get some other ones that maybe you don't like. So you might end up with a plant that's got one property that you were trying to get, but then has four other properties like slow growth or or something bad taste that, that you don't want. So undesired changes. So the molecular tools allow us to make these changes really precisely and to be pretty confident that we're only making changes where we want to make them in the genome. So a much more controlled process. And as I mentioned, when this was first done, people were doing gene knock-ins and knockouts in various ways. The zinc finger nuclease technology, TALEN technology, we're not going to talk about any of that because CRISPR has replaced all of it. Okay. So CRISPR genome editing really started in 2012. That's not when it was discovered, but that's when people started using it to do editing in eukaryotic cells. Okay. And that's the Jennifer Doudna and Manuel Charpentier work. And their first paper on this was published in 2012. Yes?
Can you kind of get into the definitions of genetically modified organisms?
Yeah. Genetically modified organism is any organism that's been modified genetically. Specifically, when people talk about GMOs, they're talking about organisms. They're not talking about organisms that have been bred to be genetically different. They're talking about organisms that have been changed through molecular techniques. Yeah, that's all I got. Genetically modified organisms, I mean, it's become a bad buzzword, but again, fundamentally what's happening molecularly is really no different than all the genetic changes we've been making really for thousands of years. It's just we can do it in a more controlled, directed fashion now. So I think it's a lot of hoo-ha over nothing, but that's just my personal opinion. Okay, so what is CRISPR? So CRISPR is gene editing that's done by taking this naturally occurring adaptive immunity system from bacteria and using it for our own purposes. What does adaptive immunity mean? Well, adaptive immunity is what we have. So when you get an infection and your body responds to that infection, so that when you see that organism again later, you have a faster and better response, that's adaptive immunity. And bacteria also have an adaptive immunity. Of course, it works in a different way, but they have an adaptive immunity against what? What infects bacteria? Bacteriophages. So they have an adaptive immunity against bacteriophages, and that adaptive immunity is this CRISPR system that I'm going to tell you about. So immunity against what? It's against bacteriophages. So CRISPR, as I said, was a big breakthrough in applying it to eukaryotes was 2012, but people didn't really catch on until 2014. It usually takes a while. So this is the cover of the journal Nature from March 2014, showing that that all of a sudden CRISPR became big news. So the world really became aware of CRISPR in 2014. And then in 2015, the journal Science named CRISPR the scientific breakthrough of the year. So 2014, 2015 is when CRISPR really started to get caught. Okay. Here's how it works in bacteria. Now, I'm going to take you through this diagram. We're going to go through it once. You'll have questions, and then I'm going to show you a different diagram, and we'll go through it again. The two diagrams are useful just for visualizing it in your mind. Okay, we're going to break this into three stages. The first stage is foreign DNA acquisition. The second stage is going to be CRISPR RNA processing. And the third stage is going to be the RNA-guided targeting of the virus. Okay. So let's start over here with foreign DNA acquisition. I remembered to bring my pointer finally, so here's a bacteriophage, and some of them look like lunar landing modules, like this one. So this looks like a T phage to me. They look like this. So bacteriophage like T2, T3, T4, T5, T6, T7. These are common kind of bacteriophage that infect E. coli. They have a bacteriophage head that contains the DNA of the virus. And when they land on an appropriate cell, they bind to a protein on the surface of the cell, and then they inject their DNA. Now, when that DNA is injected, if all goes well for the virus, the viral DNA will be replicated, the viral DNA will be transcribed and translated into viral proteins, and the virus will take over the machinery of the host E. coli cell or whatever bacteria we're talking about, and will end up killing the bacteria, and hundreds of progeny bacteriophage will be released. That's what happens if things go well for the virus and bad for the cell, right? And sometimes that happens. Sometimes the virus wins, and that's why there are viruses out there, right? But sometimes the host cell wins, and that's important, otherwise there'd be no E. coli around, right? So sometimes what happens is the virus injects its DNA, and that DNA gets cut up before the virus gets a chance to replicate and take over, right? Now, there's a couple different mechanisms by which this DNA can be cut up, and the CRISPR system is one of them. It's not the only one, but it's one of them. Now, here's the bacterial genome in a really abbreviated form. If we were looking at a real E. coli cell, so here's our bacterial cell. If we were looking at a real bacterial cell, this DNA, the genome, would fill the cell. Lots of DNA packed into the cell. So this is an abbreviated cartoon, obviously. And this part here is the part that's important to us in terms of CRISPR. So the CRISPR region of the bacterial genome is really blown up here out of proportion. It's really just a very small region of the bacterial chromosome. One of the genes, these are called Cas genes for CRISPR-associated sequences. One of these Cas genes encodes a nuclease that can cut up the viral DNA. Right? So that's one of these genes. Encodes an enzyme that can cut the DNA into 20 nucleotide fragments. So here's one of those 20 nucleotide fragments. There will be others, of course. So this is a situation where the bacterial cell wins, right? It's already cut up the viral DNA. This virus is now dead in water, right? This 20 nucleotide fragment can be incorporated into the bacterial genome. And it can be incorporated using recombination enzymes that are also encoded by these Cas genes and some other genes in the bacterial chromosome. Well, I'm about to tell you. Okay? So, the bacterial cell can incorporate one of these 20 nucleotide fragments, and so this red box here indicates the successful acquisition of this foreign DNA. So this is the foreign DNA acquisition. Now, previously in this lineage of bacteria, it could be 10 generations ago or 100 generations ago, this bacterial cell lineage acquired a purple virus. And before that it acquired a yellow virus. And before that it acquired a green virus. So it's got little 20 nucleotide sequences that it's incorporated from a bunch of different viruses. And this red one is just the most recent one. Okay, so those are the color boxes. Those are short little 20 nucleotide sequences from different viruses that can infect whatever kind of bacteria this is. Let's say E. coli, just because we're used to thinking about E. coli. Okay. What are these black diamonds? These black diamonds are the repeated sequences, the clustered, regularly interspaced, short, palindromic repeats are these black sequences here that are in between the little pieces of RNA that have been incorporated. And actually, it's more correct to say that the viral sequences have been incorporated between the repeats. So each of these black diamonds is one of the repeats, and the repeats are the same. So this is a repeated sequence. And these repeated sequences are palindromes. So they read the same forward on one strand as they do backwards on the other, like those restriction enzyme sites that I showed you. Okay? These short palindromic repeat sequences vary a little bit in size depending on the species of bacterium you're looking at, but somewhere in the neighborhood of 30 to 50 nucleotides, so not really long. Okay, so just think 50 nucleotides, that's a good estimate. So we have these black diamonds, the repeats. We've got the viral sequences that have been incorporated in between. We have these other Cas genes out here that code for the enzymes that cut up the DNA, that help with incorporation. And importantly, one of these genes encodes Cas9, or the CRISPR actual protein that's going to be involved in the cutting later on. Okay, so that's our foreign DNA acquisition. Any questions about that? That's part one. And again, this is what's happening in nature in bacteria. Okay? Yeah.
These fragments from the virus are DNA, so it has to be a DNA virus.
Yeah, this is for DNA viruses.
Double-stranded.
This is not going to work for RNA viruses. So this is a DNA virus specific protective scheme. Right? For RNA viruses, bacteria have to do something else, and there are RNA viruses of bacteria. Okay, but in fact, in bacteria, most viruses are actually DNA viruses. There are RNA viruses, but the majority are DNA viruses. So this is a really important mechanism for bacteria. Yeah?
Is there any significance to which 20 base pair fragment it actually incorporates, or is it at random?
Seems to be at random. Anything else? Okay, so that's the foreign DNA acquisition. Now, once we have this acquisition, this entire region can be transcribed. That arrow right there indicates a promoter, and that promoter can transcribe what's known as the CRISPR locus. The CRISPR locus is the repeats with the RNA, with the DNA from the viruses in between. So we can get transcription from that promoter, and here's the transcript. And the transcript, we indicate here the viral sequences with the colors, right? And the black diamond sequences are the clustered, regularly interspaced short palindromic repeats. And because they're palindromes, they can form these stem loop structures. And I can diagram that for you a little better later. But when you have a palindrome, you can get a stem loop structure. So this is the result of transcription. So this is an RNA molecule, and because it's RNA, it's single-stranded. And because it's single-stranded, it can form this internal hybridization because single-stranded molecules can pair with themselves, right? So the stem would be a region where the DNA pairs with itself, the RNA pairs with itself, and the loop would be a region where the RNA is not pairing with itself. Okay? And this is referred to as the pre-CRISPR RNA. Okay? Now, most RNAs in bacteria don't get processed, but this one does. The pre-CRISPR RNA gets processed into individual CRISPR RNA. And so here you can see now that we cleave this longer RNA into shorter ones, and the shorter ones contain the 20 nucleotide long viral sequence and contains the copy, the RNA copy of the clustered regularly interspaced short palindromic repeat. Okay? So that's the processing. Transcription and then cutting of this into the individual molecules. And this cutting is done by another Cas enzyme, another enzyme in this cluster here. So that processing is done by an enzyme encoded here. So there's a lot going on. Now, another one of these genes codes for the Cas9 protein, And that gene will be transcribed and translated into the Cas9 protein, which is indicated by this blue sphere. So the blue sphere here indicates the Cas9. So now we're moving on to stage 3, the RNA-guided targeting of the viral element. Okay. So let's take a look at this green one. The green one has bound Cas9. So this is an RNA bound to Cas9. And now if that green virus invades, this Cas9 with the green targeting sequence will match that DNA. And when it matches that DNA, it's now brought the Cas9 protein to this region, and it cleaves that RNA, and now very effectively destroy the viral DNA, or cleave the viral DNA. Once the viral DNA has been cleaved even once, that's the end of the virus. The virus needs its DNA to be intact in order to function. So once it's been cleaved, that's it. Okay? Now, you might say, well, why do we need this if we have this? Well, the answer is that this CRISPR is really efficient, and this is not very efficient. Okay? So this thing is a much more efficient system. The other thing shown in this diagram is an orange virus also invading. Well, we've got this orange one here also, and so this Cas9 with the orange fragment can find the orange target and cleave that one. Okay? So this is how bacteria use this for this adaptive immunity. It's adaptive because they're acquiring new sequences, adapting to a new virus, and then using that for defense. It would have been nice if the diagram was used a red one here so we could, but anyway, you get the idea. Okay? So this is my first version, and I'm going to take you through another diagram that shows some other features. But I want to make sure that you understand these features before we move on to the other one. So, any questions?
I kind of somewhat understand how the targets get incorporated into the genome. Is there a polymerase involved in adding the extra repeat from the other, or is there just a bunch of repeats in the genome already that you just put the fragments in?
I'm a little confused about the extra repeat on the end. Do you mean this little piece here?
So, when it's incorporating the viral DNA into the genome,
Right here.
is the repeat before that already incorporated in the genome?
Oh, those repeats are part of the genome. They're already there.
So there's a limited amount of defense.
So if you look, there's differences in different bacteria. But if you look at this clustered interspaced repeat, this was what was discovered first. So the very first person who discovered this repeat sequence, didn't know what to make of it, but discovered this repeat sequence was a Spanish scientist working in a small lab somewhere in Spain, and was studying the E. coli genome, which had been sequenced, and found these repeats and said, I wonder what this is. And actually speculated, I don't know how he had this idea, but speculated it might somehow be used in defense. I don't know how he got that idea, but it was an obscure paper published in an obscure journal, but that was actually the original identification of the CRISPR locus. So those repeats are already there. Not all of them have a virus DNA sequence between them. Some of them do, but there are others available for incorporating new sequences. So you might have 100 of these black diamonds. Right? Yeah.
You also have something called the spacer.
Yeah, so originally, when this region was first sequenced, they didn't know that these sequences were viral sequences. They saw this repeat, and then the repeat was separated by sequences that looked like random sequences, right? So they named the spacer, or they named the repeats, the clustered regularly interspaced repeats. These things they just called spacers because it just looked like some DNA that was stuck in between the repeats, right? Well, now we know that it's much more important than just a spacer, right? But unfortunately, that name has stuck, right? I love names that say what something is. This one tells us absolutely nothing, right? So,
What's the significance of the stem loop structure if that's actually what the CRISPR is encoding for?
The stem loop structure is required to bind the Cas9 protein. So this structure that the clustered regularly interspaced repeat can fold up into, that structure is what binds the Cas9. So if you don't have this thing that's folded up, it won't bind Cas9. Yes.
So when the virus was inserted DNA, it gets cut anyway. So why again? It's not a cut. It doesn't work anymore if you do one cut.
Why the virus doesn't work?
No. It inserts its DNA. It gets into fragments naturally, right?
There's an enzyme here that cuts this DNA into fragments, and then other enzymes are involved in this incorporation into the genome.
You think you mean why it inserts only once? So if there are enzymes that cut the DNA, why do you need to do it?
Because this process is very inefficient, this is super efficient. So why this one's inefficient and why this one's really efficient and why it evolved in this way, I don't know, but that's what's happened. And it obviously is a system that's ancient because all bacteria seem to do this. So it's a system that's been around as long as bacteria has been around. So they've clearly been using this for a long time. It's not as though we only find this in certain kinds of bacteria, it's found in lots of bacteria. Okay? All right. The next diagram I'm going to show you illustrates things I think. I wanted to show you this one first because it's a little more simple. The next one's a little more complicated and we'll add in some extra detail that might help. Okay? Now these diagrams are also in your manual, and I'll also have the slides sent to you. But these diagrams are in the manual. Okay, so this one's in the manual. This diagram actually comes from the NEB catalog. This one's a little more schematic, and that's why I like to show the other one first, but this one includes some detail that we don't have in the other one. So this is showing the exact same process, but it just shows a little more detail. So here's the foreign DNA that's coming in from the virus. So this diagram doesn't show the virus, but this is the foreign DNA, it's viral DNA invaded. Okay? And now here's the bacterial genome, but instead of showing it as a circle, we're only looking at the part of the bacterial genome that is this CRISPR region. That's all we're looking at. So the bacterial genome is a big circle, but we're just not seeing all of it. We're only looking at the relevant portion here. Okay, that makes sense to everybody? Here's the Cas9 gene, coding for the Cas9 enzyme. Here are those other Cas genes. And then there's a gene over here that codes for something called the tracer RNA, which was not shown in the previous diagram, but I will tell you about now. The gray boxes here are the clustered, regularly interspaced palindromic sequences. And the color regions again are regions that have been incorporated from viral infection. So the DNA acquisition again, this DNA gets cut up, and in this case, the green piece being inserted into the genome. Okay? Yeah, I'm coming to that. Okay? So far this a little more detail, but it's the same thing. It's just this foreign DNA acquisition. Now, a little extra wrinkle here is there's a sequence here called PAM, which is the sequence NGG. Now, this PAM sequence, this is specific to Cas9. Cas9, the enzyme, comes from a specific kind of bacterium. So Cas9 is not a generic term. Cas9 refers to a specific Cas enzyme that comes from a specific bacterium, not E. coli. So the bacterium is Streptomyces pyogenes. I'll put that on the board later, but it's not E. coli, just so you know. This Cas9 is not an E. coli enzyme. It's important for me to point that out because almost everything else we use is from E. coli, but this one isn't, okay? So, in Streptomyces pyogenes, what happens is that this DNA gets cleaved only if it's next to a PAM site. So the DNA sequence here doesn't matter, but it does need to have an NGG sequence right next to it. The N meaning it could be A, T, G, or C, right? So this sequence could be TGG, or GGG, or AGG, or CGG, doesn't matter, okay? But that sequence has to be there in order for the enzyme that does the cutting to actually cut. Why? I don't know, but that's how it is, okay? And that sequence is called a PAM sequence. And it would be easier if PAM sequences didn't exist. It would be easier to explain, and it would be easier to do the experiments, but that's how it is in the biology. So, any sequence on the foreign DNA can be cut, but it has to have a PAM sequence. And it's only MGG, that's only 3 nucleotides, so there are going to be a lot of PAM sequences. So it's not really limiting that much how the foreign DNA can be cut, okay? All right, that's the foreign DNA acquisition part. Any questions about that?
So this is a constant, artificial constant.
No, this is natural in the bacteria.
So the Cas9 is from Eukaryotes.
So this would be a Streptomyces pyogenes
Oh,
cell.
the whole thing.
Yeah, this would be a Streptomyces pyogenes. E. coli is doing it, but has a different Cas enzyme. Other Bacillus does it, but has a different Cas enzyme. So if you look in the literature now, Cas9 is the most common one because that was the first one used, and kind of everybody keeps doing the same thing. But there's now, there's Cas11, there's Cas12, there's Cas13, and these just come from different bacteria. Okay? Good question, yeah. We good? Ralph? Oh, another question.
So the PAM site can kind of be what it seems like, almost how enzymes will recognize restriction sites.
Yeah, it's just, a shorter sequence, right? So DPN1 has to recognize GATC, for example. Well, the enzyme that's doing this cutting has to recognize the sequence that has NGG at the end of it. So it's just a specificity that the enzyme has. Okay? All right, so that's foreign DNA acquisition. Now we can move on to the RNA processing. So CRISPR RNA processing. In this figure, it's labeled as CRISPR RNA biogenesis, but it means the same thing. I add CRISPR RNA processing here so that it would be the same terminology as in the previous figure. All right, so what happens then is that the CRISPR region gets transcribed, just like I showed you on the previous slide. These other Cas genes are going to get transcribed, not shown in the diagram. They're going to get transcribed and translated into proteins, right? This tracer RNA gene gets transcribed into a tracer RNA, and sometimes that's abbreviated TRAC rRNA or just tRNA. But they both mean the same thing, the tracer RNA. So this is an additional RNA that's produced that ends up being important. Now, here's our CRISPR RNA, and the tracer RNA actually hybridizes to the CRISPR RNA, to the repeat, okay? But not all of it hybridizes, so there's a tail that sticks off. So the tracer RNA hybridizes to the short palindromic repeat, but it also has a tail then that does not hybridize. So in fact, this RNA is a little more complicated than we showed on the previous diagram, because the previous diagram did not show the tracer RNA, right? So now we're adding in a little bit of detail that was left off of the previous figure. So each of these clustered regularly interspaced repeats will be hybridized by a tracer. Remember, these are single-stranded RNAs, so these can hybridize without any difficulty. Okay? Then it gets cut up into the individual CRISPR CRISPRs with their own piece of viral now it's RNA, right? So here's the thing after it's been cut and it's hybridized to the tracer. Okay? So now we have the individual ones cut out, each hybridized with a tracer. So that's our CRISPR biogenesis. Okay? Now we're ready for what in this diagram is called interference, or what we called in the previous diagram RNA-guided targeting of the incoming viral DNA, which is more descriptive, right? So this CRISPR RNA then gets bound by the Cas9, which was encoded by this gene. So just like in the previous diagram, you get this interaction between the RNA and the Cas9 protein. This interaction requires the tracer. So you need to have the tracer or you're not going to get the structure formed. Okay, so the tracer is critical. So now we have our CRISPR RNA, we have our tracer, and it can find the target. So here's a new incoming green virus. It finds the target because it matches. The target also must have a PAM for the cut to occur. So remember, we had the PAM up here for the cut to occur. The PAM also has to be on the target. Okay? And that's going to be important to us later. So the PAM has to be there also for the Cas9. And if the PAM's there and the sequence matches, then you'll get a cut. And now you will prevent that virus from doing the infection. So again, this figure shows the same thing I showed before, but just in a little different kind of figure and a little more detail because we added in the tracer.
So in the last step, when like foreign DNA is kind of trying to get in, because there are so many, I'm assuming there are so many of those little fragments from previous attacks, is it then like trying to fit and doesn't match, the next one goes on, fits, or it doesn't match and then goes away, and then so it's a check
so,
it doesn't fit and then cut.
right, so all these things happen sort of by random diffusion in the cell. So this Cas9 with its RNA, you know, hits the foreign DNA probably in a bunch of locations, hits there, oh, no PAM, hits there, no PAM, hits there, no PAM, finally hits one that's got the PAM and that's got it. Yeah.
So the CRISPR RNA is single-stranded. The foreign DNA from the virus is double-stranded.
Yeah.
So it needs to first pull it apart in order to be able to recognize it.
Yeah, and I'm going to show you that in more detail in a coming slide, where what the Cas9 has to do is actually separate the double-stranded DNA in order for this match to occur, and then it cuts. And I'll show you that in greater detail in another slide. So we're building step by step here, yeah.
So if these sections are able to be passed down to, let's say, future E. coli generations, is there any way to use these 20 base pair sequences to sort of see how bacteriophage are evolving, or are the sequences just too small for that?
The sequences are small, a little small for that, but you could imagine, so these things get incorporated into bacterial cell lineages over time. So this one might have been there for, you know, in the lineage for, you know, 20 years, right? And this one just got incorporated yesterday, right? During that ensuing 20 years, maybe the blue virus has evolved, and maybe this thing doesn't match so well anymore. So maybe what the bacterium then has to do to keep up is incorporate a new piece at some time in the future. So you kind of think of these defense mechanisms, just like with our immune systems, it's kind of an arms race, right? So, you know, the bacterium develops a method that allows it to defend itself, the virus evolves and changes to evade that, and then the bacterium has to respond by, you know, continuing to evolve. And so it's just, you know, the viruses and the bacteria are just constantly evolving, you know, one to try to infect better and one to try to protect itself better. And it's got to be balanced pretty good because we've got lots of bacterial viruses out there, but we also have lots of bacteria. So.
Is there any way to know how long ago a particular section was incorporated, or is that, no? Okay.
Yeah, not really. If you had longer sequences, you compare that to sequences of bacteria out there in nature, and you might be able to piece that back, but these 20 nucleotide sequences are just too short to do that.
Makes sense.
Yeah.
For the viral DNA sequences linear, so you just need to make one cut right next to the PAM sequence and you get a.
Right. So most bacteria, when their DNA invades, it comes in as a linear molecule and then will circularize like lambda does. I mentioned that earlier this morning. Usually this cutting will occur before it gets a chance to circularize. If it circularizes and you cut it once, I suppose there's a chance that things could still be functional. But typically what's going to happen is that, I've shown one green sequence incorporated here, but the bacterium might actually have, over time, accumulated 3 different green sequences. And so when the bacterium invades, it's not just getting cut here, it's also getting cut over here and also over here. So it doesn't just have necessarily one sequence per virus. It might have 3 or 4.
And the PAM is always 3 times?
The PAM is always on the distal side of the target. And that'll be more clear actually in the next diagram. Yeah.
Is the PAM site, like, random and whether or not it's important for, like, the virus, or is it just like say mutations in the PAM would then not be targeted?
Yeah, I mean, if the virus gets a mutation in the PAM sequence, so that instead of NGG it's now NCG, then that's it for the bacterium. The bacterium won't be able to cut that. So that's one kind of mutation that a virus can get to evade this system.
So this means evolutionarily if viruses have fewer and fewer double G.
Oh, right. So another way that the virus could respond is over time evolving so that it has very few GG dinucleotides. That would be pretty hard to do because GG is required for encoding, codons and a lot of proteins, and so that would be difficult for the virus to do. But for non-coding reasons, if that may occur, right? I don't know, but that's a cool idea. Sounds like paper to me. Anything else? Okay. Doing pretty good here. All right, so that's how CRISPR works in bacteria. Are we good with that? Now we're going to transition to how it can be applied in eukaryotic cells. So again, eukaryotes don't do this, right? Eukaryotes only do this if we put the right molecules into the eukaryotic cells. And the right molecules are a Cas enzyme, like Cas9, and a guide RNA. So in the natural system, this is the guide RNA, right? This is the thing that's targeting the incoming virus. So this is our targeting molecule here, it's the guide RNA. And we're going to use that terminology going forward. Now, when this is in bacteria, in the natural system, it's not referred to as the guide RNA, it's referred to as the CRISPR RNA. But when we do this in eukaryotes, we're going to call this the guide RNA. It's our targeting RNA, okay? Remember that 20 nucleotides that you put into the PCAS enzyme, or the PCAS vector rather? Those 20 nucleotides are, for our experiment, are going to be this. That's going to be our targeting sequence, okay? And instead of our targeting sequence targeting a virus, our targeting sequence is targeting a gene in the eukaryotic yeast nucleus, okay? Or if you were doing this in mouse cells, it would be targeting perhaps a mouse gene, or in human cells, a human gene, okay? So we're going to take this system and we're going to use it for our own purposes. And to take this system, all we need is Cas enzyme, let's stick with Cas9. All we need is Cas9 and a guide RNA. That's all we need. If we put in the right guide RNA and the Cas9, we can get CRISPR to happen in eukaryotic cells. And the really cool thing about that is we only need to put in two things, right, to get it to work. Okay, so let me show you that. Let's take a look at how that works. Okay, so here's a plasmid that can be used to do this. This is not PCAS. This is not the one that you're using in your experiments. I wanted to show you a different one so you can see a different example. This CAS plasmid is 10,632 base pairs. Like our plasmid, it has Cas9, right? It has Cas9. But instead of being under the control of the yeast promoter, this one's under the control of an SV40 promoter. And so, in this case, this Cas9 could be expressed in a mammalian cell. Okay. Here's an origin of replication for it to replicate in E. coli, not in mammalian cells. This is an E. coli origin. And up here is where we're going to put in our 20 nucleotide guide RNA. So the 20 nucleotides would go in right there, okay? And the promoter for getting transcription of that guide RNA would be over here. We also have to have additional RNA sequence here that encodes the rest of our necessary RNA sequence. So we need more than just this little 20 nucleotide sequence. We also need this sequence here, because that sequence is required for binding Cas9. Now I'm going to tell you what Emmanuelle Charpentier did. One of the things she did was she figured out a way to merge this sequence with the tracer sequence. So she developed sort of a merged sequence. So now this could be just a single molecule. It was essentially the repeat sequence with this tail on it, right? And she found that when she put those two together, she could get that to bind Cas9 really efficiently. So instead of having to put in this molecule and a tracer, we only need to put in one molecule. And that's why it's referred to as the single guide RNA, or sgRNA, okay? So the 20 nucleotides that you put into your plasmid is that 20 nucleotides. This sequence is already on the plasmid, okay? Let me show you that in the next slide again. This sequence encodes the repeat part of the sequence plus the tracer, and then this is the 20 nucleotides that you put in, okay? And then there's a promoter over here for expression in the host cells, okay? So you have both of these features on your plasmid. It's just yours has a yeast promoter here and a yeast promoter there, right? This one's got an SV40 promoter here and an SV40 promoter there, okay? So different plasmid for a different kind of host cell, but works the same way, all right?
Anyone using a single guide RNA?
Yeah, I think almost everybody's using a single guide RNA, because what that means, if you had them separate, you'd have to have one gene here for the CRISPR RNA, and then you'd have to have another gene for the tracer. It just gets more complicated.
Sorry, what's the promoter for the guide RNA and CRISPR tracer sequence?
So you need a promoter here to get transcription of this region to make the guide RNA. So you're not actually putting the guide RNA into the cells, you're putting in the DNA that codes for the guide RNA.
So we haven't talked about that promoter yet.
I mentioned before that in our PCAS that there is a yeast promoter here. It's not shown on the diagram, it's not shown on this diagram either, but there has to be a promoter there to get the transcription, right? And that promoter has to work in the host cell that you're using. So in our experiment, we're doing yeast cells, so it has to be a yeast promoter here. If you want to get this to work in mammalian cells, it has to be some kind of promoter that works in mammalian cells. Same thing with the promoter over here. Yeah.
In that case, like in this example, the promoters are fine, like they turn transcription factors.
Yeah, right. So all we're doing is we're putting in the Cas9 with the promoter and this thing with the promoter, and then we're gonna get expression using all of the host proteins. So, in our yeast cells, yeast transcription factors will bind to the promoters because it's the yeast promoter, and then the RNA polymerase will bind and we'll get transcription. So all we have to do is put in those two things and yeast will do the rest. Or if you're doing it in mammalian cells, your human cells will do the rest.
The production of prokaryotes in the plasma, does it, like, matter? Like,
This indicates that this Cas9 is transcribed in this direction. This indicates that our single guide RNA is transcribed in that direction. So it's just indicating the direction of transcription.
But the one that we are using is CRISPR, which is the same direction. It's gonna go around like the.
Yeah, but if we flip this whole thing over, it would still work. It would be fine. It doesn't have to be in that direction, but if you flip this over, you gotta take the promoter with it. So then the arrow would be going in that direction, and that would be okay. You can see the ampicillin resistance gene here. It has its own promoter and it's being transcribed in the other direction.
Does this plasmid matter if it's low or high copy?
Does it matter if it's low or high copy? Not when you put it into the host cell, because in the host cell it's typically not going to replicate. So in mammalian cells, these plasmids are typically not going to replicate because mammalian cells don't really do that. Yeast does, and so one advantage we have in yeast is we can get our plasmid to replicate yeast, but that's not always the case. And because replication often doesn't happen, whether it's a high or low copy number of plasmids doesn't matter so much. Okay? Where it matters is it a high copy or low copy number of plasmid in E. coli, because that's where we're going to make a lot of this plasmid in order to put it into our host cell. And so for inside E. coli, you'd like it to be a high copy number of plasmid. And so the plasmid we're working with, that PFAS, when we put that into E. coli, that will be a high copy number of plasmid in E. coli. We'll make a lot of it, and then we're going to purify that plasmid and then use it to put into E cells. Okay.
So the host cell, what is the driving force for the plasmid? You don't use nucleus?
Yeah. So when we, if you're thinking about yeast or mouse or human, you put this plasmid in. We're going to talk about how to do that later, how you get this in the cells. How does it go to the nucleus? I mean, DNA belongs in the nucleus, so maybe the cell sees the DNA and says, oh, you belong in the nucleus, and so it shuttles it to the nucleus, but it gets there. I can't look inside a cell and see exactly how it's getting there. I'd like to know, but I don't think that's really understood at this point. Inside cells, there's a lot of trafficking going on, and DNA mostly doesn't leave the nucleus, right? So chromosomes are in the nucleus, protected in there, they don't leave. But this DNA gets in, so maybe getting in is okay, leaving is not okay, right? RNA can leave, messenger RNA and ribosomal RNA and so on, but DNA mostly doesn't leave the nucleus. Okay. Any other questions I can answer? I'm sorry?
It says S1 NLS.
That's a nuclear localization signal, so there's a promoter here, and there's a nuclear localization signal. That NLS is a little tag that is going to be at the start of the protein, and that directs the protein to the nucleus. Because remember, translation is occurring out in the cytoplasm, and if we're going to get successful CRISPRing of a gene in the nucleus, that Cas9 has to go back to the nucleus, right? And so that's a targeting sequence that targets like a zip code on the protein that targets it back to the nucleus. There are different addresses for targeting different, places in the cell, right? So these NLSs, nuclear localization signals, have been known for a long time. Okay? Wow. Now, we don't need a nuclear localization signal for our guide RNA because the guide RNA gets transcribed inside the nucleus. It doesn't get translated, it never leaves the nucleus, it just stays there. Okay. All right. Feeling okay about it? All right, ready to see one more diagram?
Sorry, question.
Yeah.
There's 2 antibiotic resistant genes in what I can see.
Yeah, the ampicillin works in bacterial cells, and the puro works in eukaryotic cells. Okay. All right. Okay. This shows exactly what's happening down to the nucleotide when CRISPR works and is cutting your target in a eukaryotic cell. All right, what do we have here? This is our single guide RNA. Our single guide RNA forms the stem loop structure. And this part, all of this, so forget the N and N here, all of this is the clustered, regularly interspaced repeat part with the tracer RNA part. Remember I said Emmanuel Charpentier fused them together, and this single guide RNA actually functions to successfully bind Cas9. So this diagram shows the Cas9 only covering this part of the single guide RNA, but it's actually covering more. This is a cartoon that's not incredibly accurate, but you get the idea. So here's our single guide RNA, and the 20 nucleotides that you add is these 20 nucleotides here. So the rest of this is required for binding Cas9. This is what's required to target the gene that you're interested in. All right, here's the gene you're interested in, right here. And what happens is that this sequence can bind its match. The Cas9 enzyme separates the 2 strands of DNA so that they can actually hybridize together. And then the Cas9, if that hybridization is successful, then the Cas9 will cut both strands of the DNA. And because both strands of the DNA get cut, then that's going to be cleavage in our gene of interest. So in the case of our experiment where we're trying to cut the adenine 2 gene, you put in 20 nucleotides that match the adenine 2 gene sequence of these. When we put this into yeast, it'll find the adenine 2 gene in the yeast cells, it'll hybridize, and it'll cleave. It'll cleave our adenine 2 gene. Okay? And that's what CRISPR does. Here's the PAM sequence, NGG. The PAM sequence is on the opposite strand of the one that your targeting sequence hybridizes to. So on the same strand as the targeting sequence, it's actually NCC. It's NGG on the other strand. Okay? That's a lot to unpack. So what I'm going to do is let you go now, and I want you to think about this during lunch. Instead of doing your normal chit chat, think about this during lunch. And then I want you to come back after lunch with really good questions, okay? So I know this is a lot. It's a lot to unpack, and I'll review it again. And we're going to go through the whole experiment, and in the context of our experiment, we're going to go through all of these details, right? So, and the cool thing about this experiment is when we do the CRISPR in our yeast cells, I'm going to show you exactly how the sequences align, how things line up, exactly where the cuts occur. And then, after you successfully CRISPR and you get your pink colonies, we're going to sequence those. And you're going to see that you actually did things exactly the way that you planned, or that, we all planned together, right, to make this happen. And so that's really pretty exciting. When we get those results, I'm always pretty jazzed because that's a lot of fun. Okay, we'll stop there, go have a great lunch, and I